Hello, my name is Angel Shen. I go to Interlake High School located in Bellevue, Washington. And my project is a holistic machine learning approach for the detection of Parkinson's disease using EEG signals. Now, while I currently have my camera turned on, I will be turning it off for the remainder of my presentation as it will get in the way of my slides. So for a quick outline of my project, I will first talk a little bit about the background, such as what EEG is, some related works, and the datasets I will be using. And I will do feature engineering of my data, which leads to dimension reduction, then noise removal, classification, and also upsampling, and then eventually my experiment results. So to start, I want to first talk a little bit about what EEG is. So EEG, or electroencephalography, is the method that is used to record the electrical activity of the brain, which is then detected by electrodes placed on the scalp. You can see on the bottom left picture, and each electrode corresponds to a channel and then captures the electrical signals or brain waves, which is then displayed in the form of a time series. So EEG is usually used as a diagnostic method for neurological conditions such as epilepsy, Parkinson's disease, which is what my project will be focusing on, brain tumors, sleep disorder, or Alzheimer's. And in my project, I will investigate its potential for the early diagnosis of these neurological conditions. So before I get into the specifics of how we'll use EEG to detect Parkinson's disease, I want to first talk a little bit about some more traditional and common diagnostic methods such as MRI, CT scans, or biopsies. Now these methods usually are very expensive and there are certain risks involved that are not found in EEG. And due to these problems, many patients are either discouraged and opt out of these more traditional methods and wait until their symptoms have become more prevalent, and this can lead to delayed diagnosis and potentially more severe health problems as their condition progresses. On the other hand, EEG can provide an affordable and accessible alternative to these traditional methods. However, EEG signals are not without its challenges, as they are very prone to noise, which increases the likelihood of diagnostic errors. However, with recent advancements of machine learning technology, this issue can be mitigated, which has the potential to allow EEG to become a mainstream diagnostic method. So in order to detect Parkinson's disease using EEG signals, I'll be comparing EEG signals from healthy individuals against Parkinson's disease patients. And Parkinson's disease, it's a very common neurological disorder that has affected over 10 million people worldwide. And it affects the nerves, which then leads to movement and balance issues. So recent approaches regarding the detection of Parkinson's disease using EEG signals include these two papers here. So the first one, LAPD, they use a linear predictive coding model. And the second one, CNN, uses AlexNet convolutional networks. And these papers represent the current state-of-the-art methods, and both use very novel and sophisticated machine learning models. So the datasets I will be using, there are two of them, and both come from OpenNeuro. So I will be comparing EEG recordings from 24 Parkinson's disease patients against EEG recordings from 79 healthy patients. And from both of these datasets, patients were told to sit down, eyes closed, and rest. So here's a flowchart of my holistic machine learning approach, which highlights very key parts of my solution. Most importantly being dimension reduction, noise reduction, and upsampling, which many state-of-the-art approaches, such as the papers that I've mentioned before, they have overlooked in favor for more sophisticated models. So the first step in my project is feature engineering. Raw EEG data comes in the form of a wave, which machine learning models and techniques cannot directly use. Therefore, I've extracted statistical features such as mean, variance, standard deviation, amplitude, and energy. I've also used Fourier transform and wavelet transform to capture the frequency bands of the EEG data. And in total, I've extracted up to 15 features. So when I was reviewing the literature for this topic, I realized that many papers, including the ones that I mentioned before, after extracting their features, they directly use that data to train a machine learning model. However, with high-dimensional data, I felt I was unable to glean insights or clearly understand the characteristics of the data. As high-dimensionality data, if you were to graph and try to visualize it, the human brain just cannot comprehend this. And this lack of visibility was like going in blind, and it made me feel uncomfortable directly training a machine learning model from something I couldn't exactly see or interpret. So last year in my math class at school, I had written a 20-page paper regarding PCA, or principal component analysis, which is a dimension reduction technique. And I've decided to apply that to my current project in order to reduce the dimensions of my extracted features. So in total, I've extracted up to 15 features, meaning I had 15 dimensional data. And if you were to try to graph that and visualize it, it would be impossible. So PCA can allow me to reduce that 15 dimensions all the way down to 2. And PCA works by identifying the 2 principal components or axes that maximize the variance. And by maximizing the variance, PCA ensures that the most significant and important information from the original data is retained, and then it is then reflected in its eigenvalues. You can see this chart in the middle, the rows circled in red, eigenvalues 14 and 15, retained up to 80% of the original information. And after PCA, the data can then be graphed. You can see this graph on the left. And overall, PCA just allows for visualization of the data, and it makes it easier for machine learning models to work on. So after PCA, you can see a distinct separation of the data. However, I realized that there was still noise and outliers within the data, as indicated by the data points circled in red. And if you take a look at the axes, you can see on the very left we have negative 130, on the right we have positive 20. So these data points circled in red, they are actually stretching and warping the graph, which makes the data points on the right harder to interpret. As a result, I have applied dbScan, which is a density-based clustering algorithm. And dbScan is usually used to identify large cluster groups. However, I have repurposed it to identify data points in low cluster groups in order to identify them and review them as outliers. So data points in small, low-density clusters, such as the data points circled in red, they are considered to be noise and then removed. So after dbScan, the true distribution of the graph can be seen. However, there is still some noise that remains, as indicated by the data points in the red circles. Now, these data points, these are a different type of noise because they were mixed in with the other dense data, and that prevented dbScan from removing them. And to solve this issue, I've applied KNN, or K nearest neighbor, which identifies noise based on the surrounding points. So if you take a look at this data point on the very left circled in red, if the surrounding data points belong to a different class, so that data point is orange, but all of the surrounding data points, most of them are all blue. So therefore, because of this, that data point circled in red will be considered to be noise and then removed. After noise removal, the data is now clean and a clear decision boundary can be drawn, and I can now start classification. However, my data is still sparse in regards for machine learning models, and this can negatively impact the model performance and generally make it unstable. And to address this issue, I've employed upsampling techniques, such as SMOTE, which generates data points for the minority class. In this case, it would be Parkinson's disease. And I've also applied jittered sampling, which adds random noise to existing data points. And in total, I've generated up to 200 synthesized data points. So here's a list of all the techniques I've utilized through my project. Some of them are more statistical features, others are more machine learning based techniques. So after feature engineering, dimensionality reduction, noise removal, and then upsampling, I have finally obtained clean data which will then be used to train machine learning models. So the models that I have used in this project include Gaussian mixture model, support vector machine, neural networks, and logistic regression. So here's an overall performance comparing my approach with the papers that I've mentioned before. And I have used the common accuracy for comparison as this was a common theme across all their papers. So the data columns in the red box indicate my experiments in different training scenarios. So if you look at the data columns in green, for all machine learning models, they have all achieved over a high 96%. And these data columns specifically represent the training scenario in which only synthesized data was trained to then evaluate the real data. Now, comparing this with the papers that I mentioned before on the very left, the LEAPD approach, they have only achieved 85%, which I have surpassed. And for CNN, they have achieved a high 97.7%. However, I have achieved 98% for my Gaussian mixture model. And for their Iowa dataset for CNN, they have only achieved up to 87%. And on average, they've achieved up to 92%. However, all my machine learning models have surpassed this with over 96% in accuracy. So here are more detailed results on training synthesized data only, and then using that to evaluate real data. I've also included other metrics such as precision, recall, and F1 scoring. And if you look at the graph, all 4 machine learning models all achieved very high percentages. So here are some additional performance results in which I use the 4 machine learning models and different training scenarios. So while Gaussian mixture model GMM did achieve the best results, however, this was for training synthesized data only. If you look back on the other slide, the data columns on the very left for each graph represent Gaussian mixture model. And they generally did not do good for other training scenarios. And this is because Gaussian mixture model is very sensitive to noise. And in a lot of those training scenarios, noise was not removed. Therefore, neural networks are generally recommended for their consistency across all training scenarios. And overall, my approach outperformed existing methods mainly due to my comprehensive data processing, in which I used PCA to reduce the dimensions of my data. And by doing this, it allowed me to visualize the data, and then I could identify 2 different types of noise and then use 2 different denoising methods. So to conclude, a holistic approach with aspects such as dimensionality reduction, noise removal, and upsampling is critical for medical research, specifically regarding using machine learning techniques. And this is supported by my approach that outperforms state-of-the-art methods that over-index on modeling, and this causes them to miss out on other opportunities that I have done in my project. And my approach is not strictly for just Parkinson's disease. It can be broadly applied to other neurological disorders. And there's also potential applications in emotion analysis and sleep studies. In the future, I plan to use my approach on more benchmark datasets, as well as exploring more classifier models, and also trying my approach on different neurological disorders. So here are my references. These include the 2 papers that I've mentioned throughout my project, as well as the datasets I have been using. Thank you.